Hello, everyone, and welcome to today's DDN webinar. I'm Dr. Yuning Wang, Associate Science Editor for DDN, and I'll be moderating our discussion. We have an exciting webinar planned for you today. Our speaker, Matthew Horwitz, will discuss strategies to advance drug discovery programs with new chemical synthesis technologies. After the talk, Dr. Horwitz will participate in a live Q&A session. If you have any questions or comments during the presentation, they're absolutely welcome and it's easy to share them. Just look to the right of your screen and we will find the Q&A chat box where you can enter your questions or comments. We'll try to get to as many of your questions as possible, so don't hold back. I'd like to take this opportunity to thank our webinar sponsor, Eurofins. Eurofins Discovery has supported drug discovery research for over 40 years. The company draws inspiration from its partners to foster innovation in the pursuit of life-changing cures. They provide their experience, depth, and breadth in the form of innovative testing services and product tools that support researchers as they accelerate the discovery and development of candidate molecules. Their global team of dedicated scientists, information analysts, and client support professionals make this mission possible. Their focus drives them to continually expand their portfolio of testing methods, tools, and services to enable real, lasting improvements in human health. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section, located on the right side of your screen. Also in the handout section, you will find the certificate of attendance for participating in today's live event. And with that, let me introduce our speaker, Matthew Horwitz. Matthew Horwitz leads the Discovery Process Chemistry Department's collaborations at Eurofins Villa Pharma and manages its scale-up facility. Before joining Eurofins, he worked in chemical process research and design at Johnson & Johnson. He earned his PhD in organic chemistry from the University of North Carolina at Chapel Hill in 2017, focusing on total synthesis and asymmetric catalysis. Following his PhD, he completed his postdoctoral fellowships at the Institute of Chemical Research of Catalonia and the University of Oxford. Let's just take a moment to make sure the presentation is up. Okay, Dr. Horwitz, I'll turn it over to you.
Hello, and thank you for joining this webinar. I'm Matt Horwitz from Eurofins Discovery, based in our chemistry labs in Murcia, Spain, and today we're going to have the chance to talk about how it's possible to accelerate drug discovery using the latest technologies for synthetic chemistry. To give some context for today's talk, Eurofins Discovery provides integrated drug discovery services through a solution called Discovery One. Through Discovery One, it's possible to access chemistry services including medchem and synthesis, both with experienced project management as well as computational chemistry. Our computational capabilities expand on the classical repertoire of tools for drug discovery and include predictive AI models, as well as a range of state-of-the-art tools that allow our clients to identify potential hits faster, with techniques like virtual library screening. Our synthetic chemistry offerings include a strong FTE workforce with experience in a range of project types and modalities. We also support high containment activities including ADCs and degraders. Our chemistry collaborations integrate robotics and automation, in particular in the hit-to-lead and lead optimization phases, in order to generate targeted libraries and optimize chemical transformations. We also have non-GMP scale-up capabilities to support preclinical development, as well as GMP scale-up capabilities for IND enabling and clinical studies, with solid-state characterization support for APIs and impurities. In general, our services can be accessed in integrated format through Discovery One, but they can also be accessed as standalone offerings. To introduce myself as your speaker today, I'm Matt Horwitz, and here at Eurofins Discovery, I manage chemistry collaborations and run the scale-up laboratory at our site in Murcia in southeast Spain. Before Eurofins Discovery, I was working at Johnson & Johnson in chemical process R&D in Belgium. Earlier in my career, I did my PhD at UNC Chapel Hill in total synthesis and asymmetric catalysis, with postdocs focused on photochemistry at ICIQ in Spain and fluorine chemistry at Oxford. To get started, we're going to take a look at the stages of drug discovery in general. We can consider that there are several stages in the discovery timeline, beginning with hit finding and continuing with hit to lead and lead optimization. The culmination of these efforts is the selection of a candidate to progress to the studies associated with an investigational new drug or IND application. And while this timeline is drawn in a linear fashion, the truth is that moving along the timeline requires careful planning and efficient execution, because there are many challenges along the way. Perhaps the most persistent challenge of all, however, is time, something we cannot manufacture. For example, at an early stage you might have a small molecule which is a hit, but maybe you're thinking, how can I explore the biology of this series in a quicker way, so that you can provide concrete results to your stakeholders? This triggers further questions like, how can I get novel compounds faster? And the realization that technologies like automation need to be fully leveraged in order to go as fast as possible. Advancing through the drug discovery timeline requires being able to efficiently go around the design, make, test, analyze cycle. We start each cycle with a testable design hypothesis, which might be based on some combination of computational chemistry, AI, searching through patents and academic literature, and intuition. And then we try to evaluate the validity of that hypothesis using chemical synthesis and testing. To make that transition smoother and quicker, it's possible to use automated synthesis systems like our ChemSpeed robot that we can see here on the right, which we use for the production of targeted libraries. Once we've analyzed the properties of the molecules we've made, which might include potency, ADME properties, and off-target activity, we form a conclusion that allows us to restart the cycle with an improved perspective and a refined focus. And as we go around the cycle, each iteration ideally brings us closer to a candidate that can be advanced into clinical trials. But to get there, we have to make the right decisions, which means we have to have all the right information as quickly as possible. In this webinar, we'll be talking about exactly that: how to identify the right solutions for your drug discovery project and accelerate at every stage. We'll talk about what are the specific enabling technologies that you can use in order to put the pedal to the metal and reach each milestone faster. We'll then pivot and go through some case studies in drug discovery that will be useful in seeing how to use a chemistry collaboration to define IP spaces with novel, potent, patentable compounds. We'll also take a look at drug discovery in the context of protease targeting chimeras, or PROTACs, as well as antibody-drug conjugates, ADCs. Finally, we'll end by talking about how chemistry collaborations work at Eurofins Discovery. So to kick this off, let's start by talking a little bit more about the role of enabling technologies in drug discovery. When we're talking about enabling technologies, there's really a wide range of opportunities for both accelerating and going broader at each stage. Docking, for example, allows us to optimize the interactions that our compound has with the biological target of interest through detailed computational studies, allowing our design process to be more rational. But we also have the ability to use AI to guide our design process. With our recently developed AI tool, Sapphire, we've made a very user-friendly interface where it's possible to quickly look at the predicted ADMET properties of novel molecules using machine learning models built on large, robust internal datasets. Targeted libraries have also become increasingly important for searching new chemical space quickly in order to do hit expansion and move from a hit to a lead. In order to generate these libraries, we use the type of automated robotic synthesis platform we saw in passing earlier. Many times, though, medicinal chemistry campaigns hit a roadblock in the form of a tricky reaction or a difficult step in a synthesis. For those kinds of cases, we employ high-throughput experimentation, or HTE, to make sure that we are able to find good reaction conditions quickly. To understand the physicochemical properties of the targets being generated in medicinal chemistry campaigns, it's important to have a reliable partner for ADME as well. Minimizing the synthesis to ADME turnaround time is really critical to being able to understand the structure-property relationships in a detailed and efficient way. Furthermore, as you start to identify targets of significant interest, it's often very useful to identify a solution for high containment work, as well as scale-up operations. In our Eurofins Discovery Chemistry Labs in Spain, we've made significant investments in order to co-locate the enabling technologies that help accelerate our clients' drug discovery programs and minimize turnaround times. So where does the story of drug discovery start? Oftentimes, discovery-oriented companies will start with computational chemistry, for example, docking a potential molecule of interest into a well-defined binding site in a protein, which might have a published X-ray structure, or if not, a 3D structure generated by AlphaFold. Those kinds of approaches are represented in the top part of this figure in blue. but there's also the potential to use AI to guide the hit generation and hit selection process with ADMET predictions, which facilitate the decision-making that's associated with narrowing the broad initial group of targets down to a few series of interest. With our AI tool Sapphire, we've made it easy to go from the left side of this graphic to the right and enter into the hit-to-lead stage faster and with a clear view on aspects like solubility, metabolic stability, permeability, and other key properties of interest. Once we have a hit, it's time to think about setting up a chemistry FTE collaboration, since this is what's going to allow us to advance to candidate selection and beyond. Our chemistry FTE collaborations generally have two primary objectives. Objective number one is to accelerate the interrogation of the structure-activity relationships and structure-property relationships. This is often done by designing syntheses with diversifiable intermediates or developing diversity-oriented chemical reactions. Objective number two is to develop and execute scalable, phase-appropriate syntheses for the key intermediates and potential candidates. This is important for reaching candidate selection as well as preparing for IND-enabling studies. With that introduction on how chemistry collaborations can advance compounds through a discovery pipeline, let's take a look at some case studies. We'll also talk now about high-throughput experimentation, HTE, parallel medicinal chemistry, PMC, and scale-up. The first case study that we'll go through today comes from a collaboration with Gilead. The focus of this case study is this molecule shown below, singleferin, which is a potent cyclophilin inhibitor with some significant challenges as a potential therapeutic. It has low bioavailability, and additionally, it has 17 chiral centers and is a structurally complex natural product. Nevertheless, simplified singleferin-type macrocycles are scaffolds with therapeutic potential for hepatitis C virus. By choosing the simplified 22-membered macrocycle shown here as a starting point for a structure-based medchem campaign, it was possible to optimize the features of the series by expanding the aromatic ring system on the top and modifying the chain on the right-hand side. The outcome of this collaboration was the identification of a 21-membered macrocycle with only 4 stereocenters and high bioavailability. Importantly, the series identified in this work was novel and resulted in 4 patents, underscoring the fact that through a chemistry FTE collaboration, it's not only possible to improve potency and properties, but also to deliver valuable new IP. The second case study we'll look at comes from a collaboration with J&J. This case study is focused on regulating a post-translational modification of a protein abundant in neurons called tau, which stands for tubulin-associated unit. So it turns out that a particular post-translational modification of the tau protein, specifically installation of O-linked N-acetyl-D-glucosamine, is linked to lowering protein aggregation associated with Alzheimer's disease. Furthermore, inhibitors of the enzyme responsible for undoing this post-translational modification, which is called OGA, have potential for the treatment of Alzheimer's. In the program we'll talk about now, high-throughput screening identified this molecule as a hit, although potency and bioavailability still needed to be optimized. To do that, a chemistry FTE collaboration was started, which allowed the optimization of the heterocycle at the top, the core scaffold in the center, as well as the substituent on the nitrogen in the bottom right. In an efficient approach to the exploration of the structure-activity and structure-property relationships, SAR and SPR, different groups could be sequentially introduced to the nitrogens of the spirocyclic core using a protecting group strategy. It was also possible to explore other spirocyclic scaffolds, which are interesting both from the standpoint of lead optimization as well as covering new IP space, by identifying conditions for a challenging cyclization reaction. Being able to identify diversity-oriented reactions like this, which can lead towards many different targets in a series of interest, is key to being able to make the right decisions when it comes to choosing which series to prioritize. In general, it's also important to be able to identify which chemical building blocks are going to be most useful for quickly interrogating SAR and SPR. In this particular case, the MedChem campaign led to the rapid surveying of activity and ADME properties while scoping out new IP, which delivered the potent, orally bioavailable OGA inhibitor shown on the right. Let's now take a look at a third case study, which comes from a collaboration with Bayer focused on coronary artery disease, or CAD. There's a specific target called ADAM-TS7, which is a disintegrin and metalloproteinase with thrombospondin type 1 repeat 7 that's linked to the development of CAD. With that in mind, a drug discovery program was initiated with the idea of developing selective ADAM-TS7 inhibitors that might have therapeutic potential for treating CAD. In this case, a virtual library screen was used in order to generate the hit shown here, which was a compound from Eli Lilly that showed dual inhibition of ADAM-TS4 and ADAM-TS5 with promising potency for ADAM-TS7 as well. Here it was our collaborators that carried out the virtual screening, but this is also a key capability that we have internally to support drug discovery programs as well. At first, this might look like a relatively simple small molecule, but if you look at the stereocenter on the left, this is actually a relatively congested system, and you can quickly realize that this is not that easy of an SAR to study. To confront this challenge, the chemistry team took a diversity-oriented approach using what's called a Bucher-Briggs reaction to form the key stereocenter and hydantoin core present in the series of interest, and by subsequently removing the Bach protecting group, it was possible to reach a wide range of derivatives. By doing this, it was possible to carry out a hit-to-lead medchem campaign, which led to the replacement of the cyclopropyl ring and the optimization of the amide side chain on the right-hand side. And these modifications actually turned out to be key for getting the desired potency and selectivity for ADAM-TS7. In the end, the key outcome of the chemistry FTE collaboration was the conversion of what was initially a hit from a virtual library to the development of the potent and bioavailable ADAM-TS7 inhibitor on the right. Crucially, this derivative was very selective against a range of off-targets and metalloproteases, with the exception of ADAM-TS12. And getting an orally bioavailable ADAM-TS7 inhibitor without pan-metalloprotease activity was really the key challenge here that exploratory chemistry was able to help the project team overcome. So, having seen how chemistry collaborations can play out in practice, and how SAR interrogation can lead to novel potent compounds and new IP spaces, it makes sense to take a look at how automation is used to facilitate this process. Specifically, at Eurofins Discovery, we use automation in the form of the ChemSpeed robotic synthesis platform shown on the right to enable the generation of targeted libraries. The first step in generating a library is the library design. For this, you might already have your own ideas about how to expand on an initial hit with a library of 200 compounds, but if not, it's also possible to talk to one of our experts about what would be the best approach to cover as much useful chemical space as possible with your first library. Further refinement is always possible through subsequent iterations. The execution of the library is then carried out by an efficient reagent handling process and automation of the reactions and workups. A variety of workups and downstream operations are possible in parallel fashion, including extractions, filtrations, and evaporations. Then, the successful reaction mixtures are purified in our high-throughput purification laboratory using reverse phase HPLC or SFC. Parallel lyophilization then provides the final array of targets, ready for QC and delivery. Using this workflow, it's possible to generate novel targeted libraries in under 2 weeks, which is very efficient in terms of turnaround time. To make this process as smooth as possible, we have at our disposal over 15,000 building blocks across a wide range of categories and also a robust inventory of catalysts and reagents to enable the most common reactions of interest for SAR interrogation. This also reduces the overall cycle time, since it means you don't need to wait for procurement or reagent shipments. Let's take a look inside the high-throughput purification laboratory. Here you can see a very large lab space equipped with semi-preparative HPLCs and SFCs, staffed by a team of purification specialists to enable the isolation of compounds for the generation of these custom targeted libraries. The laboratory is organized with efficiency in mind. The compounds are first studied in screening protocols before executing the semi-preparative purifications in parallel. The analytical lab is staffed by a dedicated team that maintains the instruments in top condition and helps to accelerate analytical method development. All of our targeted arrays pass through a final quality control by HPLC before they can be delivered. A fraction of the arrays is also checked by NMR, which we have available up to 400 MHz in open access format. To see how this workflow looks in practice, let's take a look at a parallel medicinal chemistry case study. In this particular case study, a client came to us with a synthetic route to a series of interest that they wanted to explore, but it was not initially compatible with parallelization. The approach that we took in this case was to design a new route that was optimized for automation, and that sequence is presented on this slide. The scheme involved a series of protecting group manipulations and cross-coupling reactions to produce a final target bearing 3 variable building blocks of interest. In the end, a 70-membered library of novel targets was provided to the client with a 4-week production time, which is pretty remarkable when you think about how long it would take a person to go through the sequence 70 times the old-fashioned way. And really, when you have the freedom to hand a CRO a synthesis and say, make a library out of these targets, without needing to worry about whether the chemistry is already compatible with parallelization, you can start to look at a series much faster and make better informed decisions. With the number of building blocks that are commercial, many of which we already have in stock, you really have a lot of possibilities. But what if you want to start from a building block which is not commercial? Getting quick access to custom, diversifiable building blocks on scale is all about scaling syntheses safely and efficiently. We scale reactions and syntheses using continuous stirred tank reactors, which we have with volumes up to 25 liters, in order to get larger quantities, hundreds of grams for example, of the intermediates that are needed to reach a broad chemical space quickly, either with a library generation approach or medchem FTEs. In this general overview, we can see how the building blocks we choose often have multiple points where variation can be introduced through orthogonal reaction types. We also use our scale-up capabilities to access preclinical API candidates on a larger scale for toxicity, developability, and solid-state studies. Let's take a look inside of our scale-up lab. Here you can see our reactors in operation. The reactors are surrounded by a jacket with a thermal fluid whose temperature is controlled by a cryostat. The cryostats can add or remove heat, using a secondary chiller to facilitate the heat transfer. Key functions like motor velocity are controlled by an external computer, and temperature changes can be programmed and automated. We always study processes on smaller scale before performing them in larger reactors. To that end, we incorporate automated HTU workflows for reaction optimization at an early stage. At the end of a scale-up, we remove the solvent using process scale rotovaps. Moving on, so far we've been focused on how to go faster in small molecule drug discovery, but what about other modalities? I want to spend some time now talking about how we support programs involving PROTACs and ADCs using FTE collaborations that have involved high containment capabilities. In a general sense, at Eurofins Discovery we have the capabilities to handle projects in the design space of degraders, molecular glues, peptides, and ADCs. When we talk about degraders and ADCs, these are relatively modern innovations from the last 2 decades or so of pharmaceutical development. The key idea of PROTACs, which are a targeted protein degradation technology, is to construct a drug with 2 main functions. One is to bind a protein of interest, the POI, through the POI warhead, and the other is to recruit an E3 ligase, which induces ubiquitination, ultimately resulting in protein degradation. In PROTAC drug design, there's also a linker region between the POI warhead and the E3 ligase binder. All of the parts of the drug are in principle possible to optimize through medicinal chemistry FTE collaborations or also library generation approaches. When it comes to antibody-drug conjugates, or ADCs, we're talking about a modality which is, in a way, a manifestation of the dream of Paul Ehrlich, who won the Nobel Prize in Physiology or Medicine at the beginning of the 20th century and coined the term magic bullet. The conceptual framework for ADCs is essentially this. A drug linked to an antibody can be targeted to a specific cell type, for example a specific tumor, and it can interact with a tumor antigen on the surface of the cell. This interaction leads to the internalization of the ADC drug complex, which can then dissociate and kill the cancer cell through a number of mechanisms. The main idea is that since the antibody confers a very specific targeting ability to the drug, there might be a wider therapeutic window than you would have for other treatment strategies, for example, a chemotherapeutic agent. To give you an idea of what a drug discovery project workflow can look like in the PROTAC space, let's look at an internal case study which was focused on a target called PIM3. The PIM proteins are downstream effectors of oncogenes and are required for tumorigenesis. A number of PIM inhibitors have already made it to the clinic, including AZD1208, shown here. AZD1208, however, like others that have reached the clinic, is a pan-PIM inhibitor, and we wanted to aim instead for a novel and selective PIM3 inhibitor. At the outset of this project, the PIM3 AlphaFold model was used in combination with the published X-ray structure of PIM1 in order to select an exit vector on PIM3 and design a PROTAC series. The initial targets designed included motifs that could bind to PIM3 as well as cereblon, which is the protein that forms the E3 ubiquitin ligase complex. A variety of structural motifs were used in the design of each subunit of the linker, and a number of different linkers were included in the design as well. The synthesis could then be adapted to parallelization, and a targeted array of PROTAC candidates was generated. You can probably imagine that projects like this that have significant combinatorial aspects can benefit tremendously from an automated approach. This array was then assayed using our internal biophysics capabilities in order to characterize the ternary complex formation, and through this process we were not only able to generate a set of promising hits, but also provide valuable insights about what changes needed to be made for the next design cycle. In summary, even with complex, challenging compounds, it's possible to use automation to move through the assay cascade and optimize properties faster. Talking about ADCs a little bit more, here too it's possible to use the toolbox of synthetic chemistry to optimize your system. For example, by combining different structural features, you can tailor the properties of the ADC linker. In the general schematic shown on this slide, we can see an antibody and a cytotoxic payload bound together by the linker. If we look at the bioconjugation site where the linker meets the antibody, you can choose the reactive handle you want to use, which might be something like the molamide fragments shown here, which are used for cysteine-driven conjugation. In terms of the linker technologies, there are peptide and sugar-based approaches you can take which confer different, specific sensitivities. Going a little bit deeper into linker technologies, there's a problem that's sometimes encountered in ADC drug design, which is that drug linker conjugates, or DLCs, can be too hydrophobic, and this is a problem due to aggregation and a lack of solubility. To get around this, the so-called shielding linker concept was developed. The basic idea here is that by using a polar mask, it's possible to keep the hydrophobic portion of the DLC hidden. The exact way of doing this may vary on a case-to-case basis, but it can include things like oligopeptides, PEG subunits, and other moieties as well. Finally, looking at the toxin linker connections, these can be non-cleavable, cleavable, or self-immolative, and there are different possibilities for connecting the cytotoxin to the linker. Here you can see how an amino or a hydroxyl group present in a payload can serve as the point of attachment to the linker. To give a benchmark for the kind of throughput you can expect in these kinds of projects, in one ADC project we worked on at Eurofins Discovery, our chemists' efforts enabled the delivery of 75 DLCs in 6 months, each of which generally required over 10 synthetic steps to complete. When we're talking about drug discovery programs involving ADCs and degraders, it's really indispensable to have access to a high containment facility. In our high containment facility, which is co-located in our Eurofins Discovery chemistry labs in Spain, we handle ADC projects including payload synthesis, cytotoxin handling, linker attachment to payloads, and DLC isolation. We also support degrader projects with these capabilities through operations like cereblon binding fragment handling, linker introduction, and final degrader synthesis and isolation. And finally, to wrap up, I want to talk about the scope, impact, and structure of chemistry collaborations at Eurofins Discovery. Our chemistry collaborations support programs across a wide range of phases, all the way from target validation to IND-enabling studies. We have fee-for-service and full-time equivalent support from end to end, so that you can easily trigger synthesis activities in scenarios with both established and exploratory work packages involved. We've co-located ADME services with our chemistry labs to reduce turnaround time, and we've implemented the robotics system we saw earlier to speed up the early stages. We operate on a range of scales from milligram to kilogram as we transition processes from the medchem labs to the scale-up lab, and we also do process development and route scouting. Afterwards, there's a clear path to GMP manufacturing by internal tech transfer within the Eurofins network. Our global footprint offers all the capabilities needed to run a fully integrated drug discovery campaign from concept to clinic. That could mean running a hit ID campaign employing our 500,000 member high-throughput screening diversity library, executing a large-scale CompChem virtual screening, or advancing a medchem project that might involve knowledge-based approaches, or fragment and DNA-encoded library-based screening approaches, or even production of soluble or transmembrane proteins. No matter the approach, we have the solutions to advance your program. You can access over 300 chemistry FTEs in the EU and India, including our robotics lab for hit-to-lead array synthesis or to solve problematic chemistry, as well as scale-up to help your team accelerate to lead and candidate nomination. Start your program faster by accessing the broadest off-the-shelf offering of robust in vitro assays, and if we don't have it off the shelf, our deep expertise across all target classes and assay development will expedite your program. From a safety talks point of view, well, everyone knows the SERAP panel. Finally, translating your idea into understandable efficacy and a desired phenotype while gauging side effect potential can be done with our experts working in human native cells, co-cultures, 3D models, and disease tissues, as well as generating PK/PD relationships by designing and executing relevant in vivo pharmacokinetic and pharmacodynamic studies. Thank you very much for watching this webinar, and feel free to ask questions or reach out afterwards to chat about your projects.
Thank you so much for that informative presentation, Dr. Horowitz. Okay, let's get to our questions. So as a reminder to our audience, you can still drop your questions for Dr. Horowitz in the chat box at any time, even while we're talking. And with that, let's get started. So the first question is, are the yields the same when you synthesize on the large scale and on a small scale?
So I wish that it were that simple, but sometimes the yields are worse when we start to scale up a process. And we try to get a feeling for how sensitive the process will be to scaling as early as possible. So if we're really trying to aim for the best possible process, then we'll start our process optimization with a high throughput experimentation in order to really get a broad feeling for the best possible system that we can use to scale the process. And then we'll start to do larger scale validations on a few hundred milligrams, on grams. And then we'll eventually do proof of concept studies in Optimax type reaction simulators before finally transitioning into a larger continuous stir tank reactor. And it's very possible that at some stage along that process, then we can find that the yield might not be what we were hoping it would be. And then we have to decide whether we're going to step back and change something and solve the problem on smaller scale, or whether we're going to be able to push through and accept it as a sort of fit for purpose solution.
Thank you. How do the automated platforms like ChemSpeed deal with stock solution for insoluble reagents or partially soluble reagents?
Yeah, that's an interesting question. So for sure, when you try to automate any process, then there are some challenges like this that you can encounter. When you have solids, then there is a solid dispenser, which is automatable. But on the other hand, it's also possible to use slurries of solutions. For example, it's also possible to dose slurries just by vortexing them, cutting the tips off of pipettes, and dosing slurries in this way. So there are multiple options you have.
Thank you. So in the presentation, you talked about different drug discovery stages. So can you explain to us what are the main differences between early process development and late process development?
Sure. So these terms, early process development or late stage process development, I guess these are terms that are sometimes used in a vague way. But in general, when we're talking about early process development, the kind of activities that this corresponds to are things like route scouting, where you're trying to identify what are going to be the specific synthetic intermediates that are going to lead me from raw materials to API or API candidate. And additionally, we're trying to remove the risks that are involved in the process. And we're trying to figure out what are all the major issues that need to be resolved down the road. And then once we start to talk about late stage process development, then of course we're focusing a lot on cost of goods and bringing that down as much as possible, since this is often a determining factor in the success of a program. We're also talking about the environmental impact that the process has. We're talking about lowering the process mass intensity. And we're talking about lowering the amount of solvent, optimizing the amount of material throughput that we can get per unit time on a production basis, and things like this. And as we start to move through, then we focus with increasing granularity on these aspects.
Thank you. A question from another listener. How do you control the quality of antibody drug conjugates?
Yeah, so this is definitely a challenging question, but this we do with we have internal experts that handle the analytical side of this. Maybe we can follow up offline about this.
Thank you. Another question, is there a central library, or does each company has its own library from previous experiments?
Does each company have their own library of compounds?
Yeah, or is there the central library for and everyone shares?
So there are some companies that provide commercial targeted libraries for specific biological activities, but probably the most interesting kinds of libraries you can find are novel ones, which would be generated by de novo synthesis. And so if you can say that I'm interested in having 30 compounds built as a hit expansion campaign around this compound, or I want to have 200 compounds that are derivatives of this compound with a known biological activity, which would be novel in an IP space, for example. These are kinds of libraries that we automate, and yeah, when you're automating for the purpose of making novel IP space, then this is really an efficient way to build that new IP space, to add new compounds to patents, and yeah, these are probably the most interesting type of libraries to be thinking about for these kinds of collaborations.
Thank you. So you talked about chemistry collaborations at the end. Can you talk to us a little bit more? How can chemistry collaborations fully leverage parallelization or scale-up capabilities?
Sure. So when we talk about parallelization, then probably the most significant impacts that this can have on moving drug discovery programs forward as fast as possible are, on the one hand, parallelization can let us make targeted libraries, right? So we can survey SAR, structure activity relationships, and SPR, structure property relationships, much faster than if we'd be moving forward in the old-fashioned way of doing chemistry. So that's one thing. On the other hand, parallelization is also letting us optimize reactions through high throughput experimentation. And this could be by looking at different catalyst combinations, solvent, base, all the traditional variables, but just in a much faster way. And then on the other hand, we also have aspects like the ability to solve really challenging transformations. It often happens in medicinal chemistry that you say, I want to do the transformation, I want to do the route this way. And in order to make that really challenging transformation happen, that difficult cross-coupling, maybe you have to do some parallelization. Maybe you have to do some high-throughput experimentation to solve the problem.
Thank you.
We think about,
go ahead.
That's fine. Coming back to this question, the second part of this question about scale-up, this is also pretty enabling from the standpoint of accelerating the drug discovery process, not only because, of course, you'll need more material if your program is successful, but having the ability to make a lot of material, hundreds of grams of key diversifiable intermediates, this is also pretty key when you're talking about helping your FTEs, helping your full-time equivalent collaboration go faster. Because if you are able to have medicinal chemistry FTEs that are working really on the front line every day with diversifiable intermediate that's been made in a scale-up lab, then they can really do their job faster and they can get to a lot more new final compounds.
Thank you for the great insights. So another question from our listeners. How useful is a dynamic combinatorial chemistry approach at the current time in the process of hit compound discovery?
Yeah, so this is an interesting question. When we talk about dynamic combinatorial chemistry, it's tricky if we think about having lots of compounds present at one time. Of course, fragment-based drug discovery is still an active area of research for a lot of companies, and it could be that you could access compound libraries through dynamic combinatorial chemistry in this way. When we're talking about combinatorial chemistry in general, beyond the scope of just dynamic reactions, like exchange reactions, then of course, combinatorial chemistry is very important, and especially when we talk about areas like PROTAC drug design, protease targeting chimeras, and how these are often classes of drugs which are built on combinations of different segments between the POI targeting warhead, the linker, and by combinations of by basically leveraging parallelization and automation to make different derivatives that come as combinations of these pieces, then it's possible to reach a much wider range of targets faster.
Thank you. Another question that just came in. Is there a scale-up limit on the scale-up of photo-driven synthetic processes?
So when we're talking about photochemistry and scaling up photochemistry, then the solution that we often try to look for is bringing those processes into flow. And we have a number of flow reactors that work with in the photochemical sense, where we will translate a batch photochemical reaction into the flow chemical reactor. And then the optimization is essentially down to how much can we optimize the residence time so that our material throughput will be as high as possible for scale up. And this is often the most efficient way to do that.
Thank you. So what kind of recent methods in chemical synthesis are most useful for exploring structure activity relationships and structure property relationships?
Sure. So when we're talking about like chemical methodologies that are letting us really study SAR and SPR efficiently, something that comes to mind is like late-stage functionalization. Because if you can take a structure which already exists and start installing functional groups at a very late stage in a controlled way, whether that be through CH functionalization or a controlled manipulation of functional groups that may be present in the presence of a very complex molecule. That's letting us do SAR and SPR interrogation very efficiently. Other methods might include things like cross-couplings of diversifiable partners, and that could mean partners that are themselves very diverse, meaning that the cross-couplings of diversifiable reaction partners could mean cross-couplings that work in the presence of many functional groups, or it could also mean cross-couplings that provide products which are in turn diversifiable into a much larger set of final products through very reactive functional groups.
Thanks. Is it okay to have experience in multi-step synthesis of organic compounds in supramolecular chemistry to be part of the early process development, or it's just for medchem professionals?
So yeah, I think that to be part of the process to have a career in process development, let's say, then yeah, there are different kinds of backgrounds that can all be very useful. We don't need to say that training for this kind of work needs to come from one specific area of chemistry or another. We've certainly seen many successful careers in process development as well as medicinal chemistry come from people with inorganic backgrounds or from supramolecular backgrounds. And honestly, sometimes you have people that come from these types of backgrounds that are able to solve problems that you wouldn't be able to solve with just a more traditional background in organic synthesis, because they might see things from a different perspective. They might have their own solutions to analytical challenges, and they have their own mentality that definitely helps bring more diversity to the problem-solving team.
Sounds good. Another question, do you have the analytical capabilities to support IND submission?
We have, within the Eurofins network, then we have those capabilities, yes.
Thanks. And now I think we have time for one last question. So can you tell us what future trends do you anticipate in synthetic chemistry that could further accelerate drug discovery? Or are there any new developments that your team is working on currently?
Sure. So as far as future developments, I mean, some of the impacts that we've seen that have had the biggest impact and which will have the most impact are, for example, in the chemistry of radicals. We've seen the chemistry of radicals enter into a kind of renaissance in the last few years, because now we have so many new ways to generate radicals and to trap radicals with different types of reaction partners. And of course, this is very interesting from a drug discovery perspective when we think about functionalizing drugs in new ways, functionalizing intermediates in new ways to reach new chemical space. Because the more chemical space that we can interrogate, the more quickly, then the better informed decisions we can make, essentially. So on the one hand, we have radical chemistry. On the other hand, we have a lot of new methodologies being developed with biocatalysis. This is increasingly becoming an important approach for process development as well. And then as far as drug discovery, anything which contributes to the med chemist's ability to survey SAR and SPR, perhaps through the installation of bioisosteres, is also very, very useful. So there are a number of different companies that have been looking in this direction, taking a sort of building block based approach. And a lot of times when we see new drug discovery programs, then a company will come up with a lead structure and they'll start to interrogate the SAR and the SPR using bioisosteres as a way to do that. And since this is often involving exploratory chemistry, then having MedChem FTE collaboration can be super useful.
Awesome. Sounds great. Thank you for answering all the questions. And to the audience, if you have any further questions, please consider reaching out to speaker directly. His email is shown on the screen. As another reminder, the webinar will be archived on the DDN website, and you receive an email notifying you when the webinar is available on demand. On behalf of DDN, I would like to thank our speaker, Matthew Horowitz, as well as our sponsor, Eurofins Discovery, And of course, our thanks to you for listening. Have a good rest of your day. Goodbye.